I'm Barry Snow. I'm the chair of the Neurological Foundation, and welcome everybody. Thanks for coming out this evening. I'm really excited to hear our speakers, and I hope you have a good evening with us. Last night we took our staff out to Les Miserables for the Christmas dinner, and all the people on stage were wearing these, and I thought, wow, I wish I could wear them. So, I'll now burst into song. I hope you enjoy this evening. There's Professor Faull and Professor Barber are terrific speakers. I've heard them speak quite a few times, so I'll introduce them a little bit more in a moment. But we're here for both an informative and a serious evening tonight. With the pretty much control of cardiac disease and infectious disease and other things in New Zealand, countries like ours, the next big scourge is neurologic disease. And it really is becoming our health emergency, and but it's a slow boil, because stroke aside, most neurologic diseases develop slowly and creep on. But 1 in 5 of us are directly affected by neurological disease, and all of us, one way or another, would have a friend or a family member affected by neurological disease. The healthcare system in New Zealand is terrific, but it's a bread and butter healthcare system, which is what it should be. And to make things go from terrific to absolutely excellent, it requires extra support and extra engagement from special interest groups like the Neurological Foundation. The Neurological Foundation has been going for a long time now, established in 1971, and it's been funding awareness, which is what tonight is, and brain research, which is what our fundraising and science is about, for all of this time. And we see these two things as inextricably linked. We know as donors you need to know what you're funding. As researchers, we need to be able to come back and tell you what we've found and engage you in our research. So we see this as a two-way relationship. We don't receive government funding, and that's not a complaint. In fact, we shouldn't. It's their job to fund the universities, which they do really well, to fund our schools and our medical schools and our hospitals, and that's terrific. It's our job as a Neurological Foundation to represent the community, people like us and you, to put money and resources into the place that's very important for us. Because with family members with epilepsy or migraine or stroke or Parkinson's or Alzheimer's or any of these disorders, we as a community want to be able to say, this is how we want our money spent, this is where the research wants to go. And the research community is very responsive to that and actually enjoys this engagement. The thing you need also to understand about research is that it's also another slow burn. So really it takes about 20 to 30 years to go from an idea to fruition. And Ellen Barber is going to tell us about some of this tonight. And so research, you have this sort of idea of a kind of a eureka moment, you know, when you're lying in the bath and it flows over the side, but actually it doesn't work that way. Research is a long, slow, iterative process of an idea which we test, we get the results, and we test again, and we test again. And one of the very important things that the Neurological Foundation holds dear is the view that we have to be around in another 50 years' time, because it's going to take 20 years for the current ideas to come to fruition, and another 20 years for the next lot of ideas, and we want to be there to support the research community through all of that time. So we have a large part of our work as governors is to make sure that we steward the money that you donate really carefully so that it will work now and keep working in the future. And we're doing well. So in the last 18, 19 year, $4.5 million was funded into neurological research, and that's, we're very proud of that. It's quite carefully targeted money, and we divide it into two groups. And in fact, we've divided our whole granting process into two groups this last year. So we have money on pure science, and that's run by our Scientific Advisory Committee, and that's money for individual projects. So, you know, does this drug work for that, or does this piece of science work for that? But the other thing we realise is that none of this work is good without the humans doing it. And the foundation is very strong on coaxing people into neuroscience and keeping them there by funding them with studentships and fellowships and even up to professorships. And that's our and another advisory committee that we've got running, the PAC, to make sure that we choose the best candidates. And boy, we're lucky in New Zealand. We have just such an elite group of young, enthusiastic neuroscientists coming in. Their only disappointment is that we can't fund all of them. And this year we have really taken a good hard look at ourselves. Those of you at our AGM last year would have heard me talk about changing our governance process, and we are very pleased with what we've done there. And this last year, the whole staff in the foundation have been working very hard on a new branding. We realised we were getting a bit stuffy and a bit ancient looking, and we've spent quite a lot of time working on this new way of connecting with the community. And you know, there's probably a slightly older demographic in this room, but I think we've all got a smartphone, haven't we, and social media and ways of connecting and communicating that the old brand of the Neurological Foundation just wasn't working for. And so we now talk about a pathway for hope, and we feel that's going very well. Some of the big work we've done this last year is we've recommitted to the brain bank, and Richard's going to talk about this, and also continuing to fund the chair of neurology, and Alan will talk about this, as well as all the other grants and people that we're funding. I'm really proud to present our two speakers tonight, Professor Faull and Professor Barber. And I've got a very strong personal connection with this. Richard was my professor and teacher of anatomy at medical school, and although in anatomy you deal with post-mortem, he truly made the brains come alive for us. His enthusiasm was exhausting then, and it still is 40 years later. And Richard has created a brain bank which has redefined how we interact with patients and families and people who have passed on. Really, it's a world model for this. In many parts of the world, the brain bank is not so much a donation, but something taken. So you donate your brain when you die, or your family does that, and the brain goes into the freezer, and it just becomes the property of the university. And Richard has taught us and the world how actually they don't own the brains, they're just stewards for the family. And this is a gift from the person who's died and the family around them. And it's been just marvellous to watch, and humiliating, humbling at times, to watch Richard over the year communicate back with the families and keep a very strong link. And it's made the brain bank incredibly valuable because the families, because they feel connected, they're quite happy to come back and say, oh yes, you found this in the brain, well that matched this behaviour that Uncle John did. And so that's been a really important thing, and so I'm really looking forward to your talk. Alan is a great example of why we need to be here for the long haul. He trained as a neurologist in Auckland, and then he got a Chapman Fellowship, which is one of the Neurological Foundation Chapman's grants, to take him to Melbourne, where he did some really world-leading work on special brain scanning for stroke. And then he came back to New Zealand and set up a high-level stroke unit, which is actually now operating as well as anywhere in the world. And so over this 20 years or more of work that Alan's done, he has gone from basic science and neurology to now the point where with his work in Auckland, but all networked right through the country with other really good neurologists, a number of them Chapmans as well, John Fink in Christchurch, Teddy Wood in Christchurch. In New Zealand we have as good stroke care as anywhere in the world. The chance of getting the work that Alan has talked about in England is about 7 per million. Here we do 70 per million. In Scotland you don't get it at all. So feel free to collect for the stroke. We'll be here for you. Okay, that's enough of me. So, let's move on. Richard, the floor is yours.
Well, thank you very, very much for those kind words. I just hope I live up to them. They've given me a limit tonight, 20 minutes. And I told Alan, I think I can keep within that. This is a pretty big challenge for me. But the very first thing I've got to say of why I'm here is actually to say thank you to you people. Because I've met quite a few groups out there, families, and who have actually given the brains to us over the years on their mum or dad or their husband or wife or whatever. And that's the most incredible thing, which, as I tell you the story tonight, you'll see how it's transformed our research, it's transformed our thinking, It's made us in New Zealand do something that no one else in the world can do in quite the same way. And basically it's because of the community. And in our book, the patient and the family are in the centre. The researchers and the doctors and clinicians are there to pursue the research, but we're here for you see. And so tonight is, I'm saying thank you. I come out here and I never turn down an opportunity to talk to the families, to the people, to the community, because first I can say thank you. Secondly, I can tell you what we're actually doing. And so tonight I'm just going to give you an overview of how the brain bank developed and started, and how the brain bank sort of works, and then just a few quick examples of some of the incredible things we've been able to do because you people have supported us. Of course, the other support is from Neurological Foundation. And my whole career, the whole career of the development of the brain bank is because of Neurological Foundation. And you'll get to this very clear tonight. So it's and that's what, New Zealand is about teams. We do teamwork well, we work together, and Neurological Foundations like that, researchers like that, the like that, the Mr. World Cup, that's all right, we'll get it next time. So I started off in research, establishing my research career at the University of Auckland. I just come back from the United States. I spent 3.5 years on postdocs in San Francisco and at MIT and Harvard, learning the latest techniques. So you work on this little rat brain here. And you can tell there's a bit of difference between the rat brain and the human brain. And you see, virtually all research, 99% of research on the brain is actually done on the rat brain, because you can do experiments in the rat brain you can't do on the human brain, obviously. But also, there's clearly a place for doing research on the human brain. But when I came back, I'd set up my little lab at the University of Auckland, the first one doing brain research really in the medical school, and starting doing work on the basal ganglia of the rat brain. And then one day something happened which changed my life. The professor of genetics, Arthur Veal, came to see me. He came from a Taranaki family just like me, and he used to teach me at medical school of genetics and so on. And he came and said, Richard, you work on the basal ganglia of the rat brain. I look after these 400 families in New Zealand who have Huntington's disease that's caused by a gene that affects the basal ganglia. But he said, unfortunately, we don't know what the gene is, and we can't test to make sure that the diagnosis is correct for the families. And clinically, you can make a good diagnosis, but there's always an element of doubt. It may not necessarily be Huntington's. He said, the families would love you if you could look at the brain of their mum and dad after they die, to make sure and just see if the diagnosis was correct, because you see, the pathology of Huntington's is very clear. It causes a shrinkage of the basal ganglia. Massive, and when you look at it microscopically, do special tests on it, you can say this is absolutely Huntington's. So he said, would you do this? I said, yeah, sure, that'd be great. And so I kept my rat studies going. And every 3 months or so, he'd arrive along with this brain from a family somewhere in New Zealand whose mum or dad died with Huntington's, and he'd literally arrive with this formalin plastic container with a brain in it. And so we spent 2 or 3 months examining it with a neuropathologist, best clinic, and we were able to write back and tell the family, yes, definitively, yes or no. In most cases it was yes, occasionally it was no. We wished there'd been more cases. But then the family said, and they were so incredibly grateful, they were so grateful, they said, well, keep mum's brain and do research on it. And I thought, what a marvellous idea. And that was genius. Why hadn't this little country bumpkin from Taranaki thought of that? And so the families drove it, you see. And so when we started examining the brain, we found that looking at the human brain, I call it the ultimate model, we found that this Huntington's brain was somewhat more complicated than what the textbooks told us. It just didn't involve the cells dying just in this area in the same way, but there's a lot of variation. So much variation in the pattern of cell death here, and then on the outside, and I'll show you a few examples of that later on, that we were puzzled. We thought, well, why is this? And then I thought, well, let's get the history, let's get the track record of this person during life, get all the symptoms. So we went back to the families, and we started collecting all the symptoms. And we went to the doctors, the neurologists who looked after them, and we got the clinical history, we got the x-rays, and we started putting this together, and we suddenly realised that if you're going to do research on the human brain, you actually need all this incredible background of material. It's obvious when you think of it. And then we could explain that the differences in the variation in the pathology of this terrible disease was actually would demonstrate itself by the different symptom profiles, you see. And then some patients would have mainly motor movements, some would have mainly mood movements. And when we looked at the brain, we found between different cases, we could work out what the different regions did. It's like doing an animal experiment in reverse. You know, when you do an animal experiment, you sort of put a lesion in the rat brain, you see what happens to the behaviour, and then you look at it afterwards, and then you add it all together. Well, we had this brain at the end of this person's life. The family had given this most incredible gift to us. We were the custodians of the brain, and we found that the secret's actually still laying with the families. And we put all that information together, collecting the history of it, together with the clinical findings. We had a resource here for dissecting up and understanding what was going on with the disease. And so this gradually expanded over the years, you see. We've got brains from all over New Zealand, Huntington's brains. And you know, now we concentrate mainly in Auckland, Wellington and Palmerston North and Christchurch regions because of the way the mortuary services have developed and have been limited in the small peripheral areas. But we started off collecting brains from the whole national network. And we set up a donor program, and the families would become committed during life, or the person during life with the disease would write into us. And so we developed a package. We soon learned and told to the families we need to get the brain soon after death as possible. And so they would streamline procedures with the nursing home and so on. So we received the brain pretty soon after death. Some cases, the shortest time is 2.5 hours. It averages about 8, 9, 10 hours and up to 24 hours. We could go back and collect up all the history, and that gave us the secrets to help unlock the function of the brain. And so I always say that the human brain given to us by families is the most invaluable gift of science. It makes our research unique. You can't put a value on it. We are, as Barry said, we are the custodians. The family always owns the brain. We get support from Maori families too, and we treat that in a very special way. The whānau come in, very interesting, and they have a little whakanā over the freezer. And they go through and talk to their iwi, they talk to their extended whānau so they can do this, you see. And I've got to say that's humbling. So I'm here tonight as the most humble person in the world because you have enabled me and my extended research group, the Centre for Brain Research in Auckland, to do something which I could never have dreamed such a big dream. You know, my mum and dad said, you know, dream big and go out and whatever you do, make sure you're the best in the world and keep your feet on the ground and just get on with it. And that's really the secret of our research also, but they taught us how to communicate and talk to people and say thank you. You can never say thank you too much. So over the years we've expanded not only Huntington's but Parkinson's, Alzheimer's, epilepsy, motor neuron disease, MS, glioblastoma and other rare diseases, but those are the main ones. And the Neurological Foundation, you see, well, we got the first brain in 1980. By 1994, I realised, 1993 actually, I realised, my gosh, we've got 60 odd brains. And I said, yeah, we've actually got a brain bank going here. Why don't we call it that? And we're having real problems funding it. So we went on to see Dr. Philip Wrightson, and who headed up the Neurological Foundation at the time. And I actually used to be his house surgeon when I was doing neurosurgery. So we had a great relationship, and he was wonderful. And I said, how about we name this Neurological Foundation Human Brain Bank? You think the foundation would fund it? He said, oh wow. He said, put it in a letter, so a 2-page letter, wrote it in, and the rest is history. So this is our Human Brain Bank. This is the room where we dissect up the brains there. We are a world-class resource because of the way we dissect the brain and carefully process, and I'll get into that in a minute, but also because we get the track record, we get the history of the brain we're looking at. Now in New York, they've got a huge, marvellous big brain bank there. Professor Jean-Paul von Sertel, world authority on Huntington's, he runs this brain bank, and they get brains from all over the United States. The time after death is you know, it can go up to 2 days sometimes, and they process in they slice it up and they slice it and they freeze it down. They don't collect they do the pathology of it and say this is a Huntington's or Parkinson's or Alzheimer's or whatever brain. But they don't get the patient details, because you can't do that in a country, the size of the US. You can do that in New Zealand, and that's what makes us our resource, a world-class resource. So when we receive the brain, you see, we used to only look at the inside of the brain, which is the basal ganglia, which was the main reason we thought was involved in Huntington's. We found that other areas were. So we gradually developed over 25 years, 30 years really, a process of just taking the brain apart in a Legos type way, you see, blocking all the individual gyri, we give them special names, I won't go into all those details, and so we get 60 blocks from this side of the brain, 60 blocks from the other side of the brain, and the net result, you see, this is what it looks like from the medial side of the brain, and so about 120 blocks roughly per brain, so that gives us, we've got over 45,000 blocks in storage, and it's tissue from actually 1,000 brains. This is regal material, this is material which we know a lot about. And when we ask the families if they would be happy, sometimes there's only a certain amount of research we can do on the brain in New Zealand. And through talking at conferences, we connected up with leading research groups in the world who are doing different things, and they're working mainly on animal brains, and they say, listen, can you send us a little bit of tissue so that we can check up that saying what we're seeing in the rat brain is actually true, and we can do our special techniques on it, whether they're gene studies or whatever, chemical studies, biochemical studies, and we can do, we'll do that, and we couldn't do that. We didn't have that sort of resource, we didn't have that sort of expertise. And so we could do that as long as we worked in a collaborative way through our ethics that provided us to do that, as long as they were actually had to be identified as extensions of our lab. And that was great. And when we asked the families, were you happy for the send a bit of mum's brain to Zurich or to Oxford or whatever? In every single case, they are delighted. They said, mum's getting the travel and death that she didn't have a life. Or dad's contributing to the future of our kids. His brain is living on and used for research to give our kids, our family, a better future than what we had. I've got to say, so we stay in contact with the families forever. We can talk to them. They can ring us up anytime they like. And we often have to go back and talk to them. Often they will come, and now and again. I had a family a couple of years ago who were all getting together for the Christmas Day dinner, and their dad had died of Huntington's, the previous year. They said, we've got all the family here, and we just want to know. And they just want to know how research is going. Well, it's now on the phone, the best hour of my life, in a way, just telling them and giving them hope for the future. And since then, that research has moved on for Huntington's, and we're now trialling a way of how we can try and turn that gene down. So it's marvellous to be able to tell them that is the most wonderful thing in the world. And so you see, this is what it's a partnership with families, a partnership with doctors, a partnership with the brain scientists. And the most interesting thing, which we never knew, is that we got unexpected findings. I'm going to show you a few slides. These are just snippets, all right? I'd love to tell you more, but I'd be here all night. So things we never expected to find. For example, you may have already heard that we ended up finding a pathway for new brain cells in the human brain. This is the work that Morris Curtis and other PhD students did in our lab, and we were told there's no such thing in the human brain, so we now know that we can make new brain cells forever. We've got to work out exactly how we can promote that and how much repair you can really get. But this suddenly turns us and means the brain is plastic and it's changing continuously. And we got that published in Science, which was would never have dreamed of a publication in the top. Well, Science and Nature are the two top journals in the world. We're on the front cover. I'm always selling one, it's wearing a bit thin, I know, but it's actually better than being on the centerfold table, actually. And then looking at Huntington's disease, we found that initially, you see, we thought that it was just a disease of the basal ganglia. And we not only found this is highly variable, we also found that there was lots of degeneration in other parts of the cortex. And literally 10 PhD students, we systematically over 10 years looked at all the cortical regions. That was a mammoth undertaking. Put the whole picture together. What it showed is that patients with mainly motor symptoms or mood symptoms or mixed symptoms would show patterns of cell degeneration which were quite different, and therefore we would be able to work out what parts of the brain were responsible for those symptoms. So we were beginning to understand the brain and also the complexity of the disease. And the latest finding we just published is showing that in Huntington's disease, there is a region at the back of the brain called, which is the cerebellum, a huge region, and we were told that's the normal part of the brain. In Huntington's, it's not affected. Well, we've just shown that, in fact, in cases which are mainly motor symptoms, there's a significant cell loss. You know, a 31% loss of Purkinje cells, which has never been realised. But in cases which didn't have motor symptoms, mainly mood, there's no loss. So that showed again the complexity and the fascination of this incredibly. We thought it was a simple disease where one gene caused a pattern of degeneration. That was it, the same for everyone. But no, it's variable. And so that gives us a better scientific understanding. It means we've got to change our models and make sure they are right. The other thing which has been incredible is that because we get the brain soon after death, because the families support us in this most special way to make sure we receive the brain, and also because we get tissue, so every patient that comes into Auckland City Hospital and is neurosurgeons operate on, when they remove tissue, we have consent from the patient and the families that we can use that tissue across the road in the Centre for Brain Research. We take it over and we can keep brain cells alive and grow them in a dish. It's quite a complicated process which took about 5 years to develop. This is the work which the Hugh Green group developed, and some of you may have seen recently in the last few days that we've just got a $16.5 million grant to take all this work to another level, and that's just incredible. So you see, we can actually take small pieces of tissue from the brain soon after death or off our brain total, make cell cultures, keep these brain cells alive. And so we have, these are cells which look from a healthy brain, and the cells have been separated. You see, this is not how they normally are connected, but you can do a lot of things by looking at cells like that, and looking at disease cases. You can then study these individual cells. It's like a disease in addition. Ask these human brain cells from an epilepsy, Parkinson's, Huntington's, or a glioblastoma, a brain tumour cell, and you can ask them questions and test new drugs against them, and it can enable just take you to another level. By doing this in the human brain, we're developing a better understanding for the human brain and disease, and we're developing a therapy in the dish. See, and that's what the Hugh Green group actually funded. Now, I don't want to read all this, but just to show that we can virtually culture every type of glial cell and also blood cells, and even we can culture neurons, you see, and keep them alive. Now that was unheard of, and if I'd said this 10 years ago, I would have said crazy, you know. The Hugh Green funding has enabled us to take what the Neurological Foundation has given us in terms of providing this tissue. They've taken it to another level. But you see, if we didn't have funding from the Neurological Foundation to have this brain bank, we couldn't do this imaginative research. Now, no other group in the world is doing research like this on the human brain in this way. So this is where I say, you know, if you're a small country, can sometimes do things better than a big country. And over the years, we have developed collaborations, wonderful collaboration with the leading research groups around the world. And what's interesting, there's so much competition between these groups. They wouldn't collaborate with each other, but they would collaborate with us. and so we're sort of the brokers of international collaborations. And I got to say, my life is the most wonderful life ever, and I get paid to go to work, and my wife's to see God. So it's a partnership. And so I just want to end, this is my last slide, and say thank you to the Neurological Foundation for helping us have liftoff on this exciting rocket, you see. But of course the rocket could only be launched with support from the community, so thank you very much.
Well, isn't that great? One of the crosses I have to bear is following Richard often, and Richard also used to take me to the anatomy. Used to have one-on-one sessions when I was a first-year trainee. I don't know if you remember that, Richard. always fantastic. So, change of tack. I'm going to talk about stroke, and 1 in 4 of us are going to have a stroke at some stage. This is a big deal, and it's the second biggest cause of death after heart disease, and it's the major cause of long-term adult disability. In this country, a quarter of people are under 65, they're at working age. And in hospitals like Wellington, Auckland, Middlemore, there'll be someone in their 20s once every 3 weeks, 4 weeks. There'll be someone in their 30s every fortnight, and we get, in Auckland Hospital, we get 3 or 4 people in their 40s every week. So it is by and large a disease of older people, but it affects a lot of young people. And unlike the work that Richard does, it's about plumbing. So I'm a plumber. And so this is, I'm going to take a test at the end of this. So watch this. This is the brain. These are the arteries in the brain, like a tree, supplying blood to different parts of the brain. And this is a clot. It's come from the heart or come from an atherosclerotic plaque in your neck. And it goes up, and at some stage it's too big to go any further. It gets blocked. It blocks the artery. And what happens is the blood stops flowing, and the brain immediately downstream stops working, and within minutes it'll start dying. Okay, and it doesn't come back. But all this tissue further out, it's got reduced blood flow, but what happens is you're getting collateral flow from these other arteries. They're trying to keep this part alive, and that's called the penumbra. Okay? And at some stage, the penumbral vessels fail. It's just not enough, and the brain will start dying, and this infarct core gets bigger and bigger and bigger, and will eventually expand into most of the full extent of that hyperperfused area. So this is what happens in stroke. And it is plumbing, it's not rocket science. This is a catheter. This is the same artery as I just showed you, this one here. And this is the catheter. You go in through the groin, you feed up a little catheter, and you squirt dye. You take x-rays, and you can see it. Can you see it? And you come up, anterior cerebral artery, and it stops. And you can see here, this is the skull. There's a big wedge of brain with little or no blood. And unless you open up that clot, that brain's going to die. And the treatment that we have had until relatively recently is Alteplase. You give it into the vein, and it's effective, and you're 30% more likely to have a good outcome. But it's not good for these big clots. Okay, and I'll show you a clot in a minute. You'll see what I'm talking about. We can see now with the fancy scanning techniques that we do, we can actually see that area of brain with reduced blood flow. Okay? So there's a clot here, and we can see this reduced blood flow. And we can actually watch it as the infarct core. We don't like to watch it, but you can actually see the infarct core growing if you do serial scans.
Could you explain the colours?
Oh, the colours are just, this dark stuff is where there's reduced blood flow. That's the bad. So this is the normal side, that's the bad side. Okay, so half of this, not half, but you know, a good significant proportion of this person's brain is not getting any blood. And so the Alteplase, the drug that we give into the vein, is modestly effective, but it only opens up these big clots about 10 to 30% of the time. And so we were involved in one of the trials. We've been trying for years and years to figure out a way of pulling these clots out, and we were involved in a trial run from Melbourne, which is where I trained, of this little solitaire stent retriever. And we feed that in, and you feed it into the artery, and you go through the clot. That was the trick that we didn't realise. And you actually open the stent retriever, it's like chicken wire, from the end back towards you, and it enmeshes the clot. We were also tried it with a corkscrew from down here, trying to get into the clot, but you just broke off bits of the clot that floated downstream. Didn't work. Okay? And then you can convert this patient from this to this, the same patient. This is the before, this is the after. Okay? Now some of this brain would have already died, but the trick is to do it very quickly before too much dies, and this is the clot. That's a fingernail, that's the thumbnail, so that's I don't know, it's huge. It's not a surprise that the drugs that we were giving into the vein doesn't always work. So we're providing this treatment. We provide it across New Zealand. There are 3 centres. There's one here in Wellington, Auckland and Christchurch. These are helicopter flight times. So we've piggybacked on the cardiologists who've been flying people around for doing the same sort of stuff, stenting coronary arteries. And so we've piggybacked on their helicopters. And so these are the flight times. Taranaki, the first patient from Taranaki, we got a call, and I thought, I got my little map out, and I thought it was 2 hours from Taranaki. You know, sat down, had some dinner, and was waiting, and got a call saying the patient's here. I didn't realise Taranaki's got a fast helicopter. And they were there in an hour. And these are the numbers of clot retrievals that we've been doing. So we started in 2011 in Auckland. This is when the trials were published, and you can just see it's exponentially increasing across the country. And as of a couple of days ago, we've done 350 patients in the past rolling 12 months here in New Zealand, which is amazing. I've just been on a sabbatical in the UK, and we're currently treating 70 million people across the country. The UK is treating 7 million people across the whole of the UK. And so we're further ahead than I'd actually realised. This is the outcomes, okay? So what happens to these people? At 3 months, 40% of people are almost alive. Almost alive. Almost normal. Almost alive. No, 40% of people are almost normal. Normal or almost normal. So not that you would notice, basically. They may have symptoms, but they can do everything they could do before. If they were a violinist, they can play the violin. 50% of people are independent. They can look after themselves. And remember, these are the biggest strokes you can have. So these are amazing figures. 68% of people end up at home. Okay, so this is a transformative therapy. I wouldn't have thought I'd see this in my practicing life. Now, what about research opportunities? So this is wide open. This is a new treatment. We're at the forefront of providing it. And there are lots of research questions that we don't know the answer to. Okay? So it's not a done deal, that we're still finding our way with this treatment. And so over the last 9 years we've been doing it in Auckland, 8 years we've been doing it in Auckland, we've got a big database of people we've collected the information on. And the foundation has very kindly funded a clinical fellow, and Will Dyprose has done that this year. And I'm going to present some of the work everything I'm going to present now is work that the foundation has directly funded. And these are some of the questions we've looked at and answered, and it's been really exciting. So one of the things when a person comes in is you want to know, is this person going to do well or not? Is this the sort of person that you should be treating or not? Now that sounds like a difficult thing to think of, but these are the sort of things that, as doctors we do. Are we going to cause this person more harm than good? And so some of the things we've looked at in terms of prediction is, not surprisingly, the more brain atrophy you've got. As we get older, we lose brain cells and we've got a little bit of shrinkage. And more shrinkage is bad, and that's probably not a surprise. But what Will did with his brother, who's a computer whiz, is develop this technique to automatically determine the amount of brain atrophy. You know what I'm trying to say. Automatically, from the scan as they came through the emergency department. So this is an automated technique, and basically they set up this computer program. That's the skull. They identified the CSF fluid. This is the fluid that surrounds the brain. And by doing that, they could measure this volume of CSF as a way of getting an indication of how much shrinkage a person has. And not surprisingly, the more shrinkage you've got, the worse you do. The next thing they looked at is developed an automated program to determine this. This is the back of the brain. This is back here. So the person's lying on their back. This is the cerebellar hemisphere. These are the cerebellar hemispheres Richard was talking about. This is one of the vertebral arteries. This is one of the 2 arteries that supply blood to the back of the brain. And you can see this one because the wall is full of calcium. It's almost bone. Can you see that? And this is the blood on the inside of it. And Will and his brother have developed a program to automatically determine how much calcium you've got in your arteries, and have published this. And this is how bad having calcium in your arteries is. This is the modified Rankin scale score. 6 is dead. And this is if you've got calcium in your vertebral arteries, and this is if you haven't. If you've got calcium in your vertebral arteries, and it's not that uncommon, your chance of dying after clot retrieval is 60%. If you don't have it, your chance of dying is about 18%. Okay, so that was a very interesting finding. What else have we done this year? This has all been done this year since January. So we started on December the 20th last year. If you've got high glucose, you're going to do badly. If your temperature is increased, even within the normal range, the higher your temperature is, for every 1 degree increase in temperature, you've got a greater chance of having a poor outcome. And so what we've been doing is we've been developing all these steps. We've got a big database. No one else has done this yet. And the plan is to develop an way of just putting all these variables when a patient comes through the door, because we collect these variables, the nurses do as we go. How long ago was the stroke, what sort of the stroke, and then automatically put this, what their glucose is, what their temperature is, and have an algorithm that automatically tells you, is this person going to do well, are they going to do badly, what's their chances of doing well, what's their chances of doing badly, to help us make decisions and help families make decisions about whether or not to treat. The other thing is the contrast agents. So this fancy scan I showed you, you need to give an injection of a contrast agent, which is basically a dye. When you're doing the angiography to do the clot retrieval, this black stuff is dye. You're squirting dye, taking an X-ray. You have to get rid of the dye. You have to excrete the dye through your kidneys. And the dye, the contrast agent, can damage your kidneys. And so people have been concerned, are we causing kidney damage by doing these fancy scans and by doing the clot retrieval? And so we've looked at this, and in fact, the chance of having kidney damage from clot retrieval and the scans you do beforehand is 1 in 33, so it's about 3%. So significant kidney damage just from doing the procedure. If you do have kidney damage, it's not good. But we also found that even taking this into account, doing the clot retrieval is the best chance the person has. Okay, you can fix the kidneys later. You can't fix the brain later. The other thing people don't know is, what do we do? How do we do this? Do we sedate people or not? So these are all the research questions we've been doing this year, and there's been a huge debate about this. And the options are, you can do clot retrieval under conscious sedation. And so I've just spent time in Calgary, and the neurologist goes in and sedates the patient. And the patient's awake. They can follow commands. They can, lift up your hand, do this. And a number of studies have suggested that doing it under conscious sedation is better. People who have it under conscious sedation have a better outcome. But the other option is doing it under general anaesthetic. And doing this is much easier if the person's not wriggling around. And people wriggle. It's uncomfortable when you pull the clot out. It actually hurts. And even though it's on the inside in the brain. And you can also control a person's ventilation rate, their breathing, very carefully. And so there were a number of small studies that looked at randomizing people to conscious sedation or general anaesthetic, and they were all negative. But they're all done by anaesthetists. And there's no anaesthetist here. And the endpoints they chose for stroke trials were ridiculous. I mean, they were anaesthetic out endpoints for people with stroke. And so we've taken the data and we've done something called a meta-analysis, put these 3 trials together and actually looked at neurological outcomes, the sort of outcomes that you do in a stroke trial. And we found that general anaesthetic is clearly better. Patients have a higher rate of actually opening the blood vessel. And if you don't open the blood vessel, the patient doesn't do better, and that's because the patients aren't wriggling. you've got a greater rate of independence at 3 months. And these are not subtle improvements. The number needed to treat under general anaesthesia is 8. For one more to go home independent compared with doing clot retrieval under conscious sedation. That's a major benefit. And so that's one of the other things we've done this year. The final things that we have looked at is, are there a difference between the general anaesthetic agents we use? You can either give it by an injection or by vapor. They're not all the same. And to cut a long story short, we've shown that it's better to do it under propofol, which is an injectable one. And the last study that we've done, or as we're about to do, in fact we had our first patient recruited yesterday, is something called master stroke. And this is what blood pressure do you maintain in patients when you're doing the clot retrieval? Do you keep it really tight? But if you keep it really tight, you might damage those collateral vessels I showed you at the beginning. And so the infarct may increase. But if you are permissive and the blood pressure is higher, there's a greater risk of bleeding. And no one knows what to do. So we started yesterday a trial that's been funded by the foundation, and we're randomising people to a permissive, we're not too worried what the blood pressure is as long as it doesn't go sky high, versus a tight control. This is very exciting. There are a lot of people that are waiting for the answer for this trial. So these are trials we've done this year. All of these studies we couldn't have done without our fellow, Will Dyprose, who's a machine. He's published 6 papers since January. To put that in perspective, in my PhD, and I thought I was pretty good, I published 5 in 3 years. So he's been the machine, and the foundation's funded him. And my colleagues, Doug Campbell, who is a neuroanaesthetist, Stefan Bruin, the radiologist who actually do this, and the neurologists whose sleep has forever been disturbed by doing this. But finally the foundation, because without the support that you give, this wouldn't have happened. The foundation funded me as a chaplain to do my PhD in Melbourne. The foundation has funded me consistently over the years to do research, individual studies. It funds the chair, it funds Will, it funds MC Smith, who's the academic physiotherapist. This research, particularly the research I've just presented, wouldn't have happened without your kind support. So thank you.
Okay, thank you. I was just sitting there reflecting that none of this work gets done without academic neurology, and which is the difference between standard healthcare and the next level up. And I was thinking that actually four of the leading people leading the stroke research in New Zealand all have chaplain fellowships from the Neurological Foundation and got them going down the path instead of just straight clinical neurology, been doing clinical research.
Yes.
And I just wonder about the two of you, because you both did this, didn't you? You both started out your lives kind of heading down the standard path of being a standard doctor, which is pretty good, but you changed tack. And I'd just be interested to hear, Richard and then Alan, about how that happened and how we get the next generation of young researchers going.
Well, so when I went to the United States, I was on a Harkness Fellowship and a MRC Fellowship at the same time. And I needed a little bit more money actually to finish the work off in Boston, so the Neurological Foundation helped me out. And that was when the foundation was quite small, and I could write back to my colleagues back home, and they would just, in an ad hoc way, just say, yeah, we'll take this risk. And when I came back, of course, at every step along the way, we got research money from the foundation to pursue looking at, what was going on in Huntington's disease, and in cannabinoid receptors and so on. And then, of course, when we got the funding from the Brain Bank in '94, we put it together, and then it's just developed now. And because, and I'm sure it's the same for Alan in the way, that your research road, you would like to think, graduate students come and ask me, I want to plan my career. And, can you help me? And I said, yeah, I can help you. I said, first thing is don't plan your career. But I said, take every opportunity that comes up, and a door is there, and just close your eyes and just go through. because coming out the other side is something completely different and a different opportunity. And I said, it's a bit risky, but you've got to take risks in this world. Now, the Neurological Foundation has sort of given us funds to do that, you see, and in the most imaginative way. So we wouldn't have developed the diversity of researchers. You know, we have across the Centre for Brain Research, we've got about 450 people. There's about 200 odd graduate students. Many of those graduate students have got different fellowships for their PhD or their postdoc or whatever, gone overseas or another neurological field. And they have invested in the leaders of tomorrow. And they are the leaders of it. So I'm stepping back from doing lots of things and making sure that others come up. And I've got to say, we have got the talent of the next generation of leaders. It leaves us all for dead. They've got to have support so they can take risks too, you see. And so the brain bank was sort of it happened, but we got support from the foundation to continue and expand it. Doing these other areas of research and Huntington's and other things and all the rest of it, that we got support from the foundation often, which we couldn't get from other sources, but it was it provided a generation, ongoing generations of young people being able to pursue their dreams in different ways. The opportunities now in research with the fancy different microscopes they have, the different imaging systems with psychologists looking at brain function of people alive and doing wonderful studies working out what parts of the brain do what. It's incredible now, it's just expanded hugely. And so it's another generation. So I don't know whether that answers your question. But it's...
you don't have to say Neurological Foundation, but...
Well, I mean, you know, the fellowships the foundation fund, you get a young, keen scientist or a clinician, you know, a neurologist or a neurosurgeon, and you can go anywhere in the world and say, can I come and work with you? You can go to the best places in the world because you've got funding, and they always say yes. And so the clinician, you're a chaplain, the clinicians in New Zealand, the neurologists and the neurosurgeons are amongst the best trained in the world because we've had that opportunity. Were you a chaplain, Tim? No, I wasn't. No, okay. But you know, you can
second best is the best. Keep going.
But you know, you can go anywhere in the world and you can say, can I come and work in your lab or can I come and work in your unit for a year or 2 years, and that's just been fantastic.
All right, now, time for you folks to start talking. Questions for the panel?
And I'd like to know how you arrange to have your brain go to the brain bank.
Very easy. Best thing is just Google Centre for Brain Research, and that comes up the donor, the process for actually donating a brain, and we'll just write to Centre for Brain Research, University of Auckland, and that'll get us, and we'll follow up. And we'll call you and talk to you if you need to. We will then send out a package to you which you can read through, and we've got staff who can then talk through and tell you a bit more about the process. So just you, mate. So we always wait for people to approach us, and then we'll follow up with you individually.
Are you only interested in the brains of people with a neurological disease?
Great question. In fact, the biggest problem we have is getting, every time you look at a diseased brain, it's important you have a non-diseased brain to look at. And so a person who's died from a non-neurological cause, so we call them sort of normal controls, if you like. We have more problems getting those controlled brains than the diseased brain, because people with a disease, the person or the family are very committed to helping this disease. And often we get, actually, what's really interesting, we get is people from the family, they will also become donors because they will may not die of a neurological disease, and we just point out to them here, that's actually critical for us. So you know, we desperately need more normal brains, and people just enroll, of course, and you can't take the brain until they die. It's the brain. That's the way life goes. We love donors, but we want to make sure you have a great life. And so it's so we actually get we are well supported from the different diseases generally. We can only accept about 25 to 30 brains a year because of the very rigorous way it's high intensive work. There's 8 to 10 people involved each time we receive a brain. It's like when the fire, the fire station, the bells call and all the team comes out. Well, same when we receive a brain, the bells go and we have teams which are in rotation. They come out and they help with the permanent staff, and it's all students. So and to do the processing, which is very time intensive. So it's about 35 brains a year maximum, but we need a significant number of those to be normal, and that's where we need support.
Perfect. Another question just there.
Hi, my name is Marina. First of all, thank you for your talks. I have a question for each of them, if I can. First question would be for the clots. Is there any way to take the clot out before the stroke actually happens? Like if you detect it in some other tests and you see that there's a blood clot and you can take it before it actually impacts?
So the cardiologists, lots of the clots come from a condition called atrial fibrillation. Instead of the heart going blood dub, the atria fibrillates. And blood can clot on the inside. And so about a third of these strokes that we do clot retrieval on have got atrial fibrillation, and by using medications, we used to use warfarin in the old days, but there are newer ones now, to actually thin the blood, we can prevent those strokes from happening. And we can prevent probably 70% of the strokes happening. It's something that we need to look at because many of our patients have got atrial fibrillation, they knew it, and they weren't taking their medications or they weren't prescribed their medications.
That's good news, thank you. And another question would be for you, Doctor. Are there any biomarkers that we could detect in blood? For a specific question is that I worked on the platform technology for really extremely sensitive diagnostics of different proteins, and I would love to combine that with diagnostics for some neurological diseases, so if there's anything that we could focus on.
There's a huge research effort going on by a team actually across New Zealand. We've got a centre of research excellence called Brain Research New Zealand, and we have developed under that umbrella the clinics, dementia prevention research clinics, where we have enrolled patients and people who are in the very early stages of Alzheimer's disease. And one of the things we're looking for in those cases is to identify markers which we may be able to use to monitor as the disease develops. Can we find markers which are changing, and therefore when we develop a new treatment, which we're trying to do, can we then see if we can keep those markers at the low level and hence prevent the onset and slow the progression of the disease. Now, doing that in Alzheimer's, Parkinson's, Huntington's, this is a common challenge, and so that means doing research, collecting bloods from these patients. And Tim knows all about this. He's directing and heads up the arm of the Centre of the Brain Research New Zealand, looking at cases like that in Parkinson's disease and so on.
Perfect. We've got another question. Ada?
Do you work with children, or do you take the brains of children on time? And how do you set an age limit for the sort of downward level that you take brains for?
Well, in special cases where we get consent, we can accept, and we do accept cases of, paediatric cases. We don't get the same. The network we have established is very much for the, going from, we've got cases from age 20 up. Most of it's in the sort of mature age. We have looked into this, and we're seeing there are, because just the way the systems operate in New Zealand, we have to try and overcome a few challenges there. Often it's the last thing they're thinking of is brain donation, they're thinking about other things. But we're looking, because looking at some of the causes of, the types of tumours and so on, which early in life is very important too. So that's on the agenda, but we haven't really fully developed that.
I just want to thank everybody tonight, and I think You know, the new thing of the Neurological Foundation is a pathway to host, and really tonight has been about that. It's also been a lot of talk about gift tonight, isn't it? The gift of brains, the gift of the money that goes through the foundation to do research. And a couple of Christmases ago, I was on call on Christmas Eve, which meant I had the phone, and I was a little bit cross about that actually. You know, didn't get to go to Christmas carols because I was called out, and I do enjoy a little Christmas drink, and I wasn't allowed that either. And then 2 in the morning, I really was frustrated because I got a call. I had to do one of these clot extractions. As a 34-year-old from South Auckland, he had just had a new baby, and he was paralysed down the left-hand side. And they rushed him across to Auckland Hospital, and we all went in. It was about 2:30 by now, and I was very dark on this. And we did this whole procedure that Alan described. And as we woke him up on the table, his paralysed arm, he lifted it up like this, and he waved to us. And the anaesthetist turned to me and she said, you know, in all my years of medicine, this is the greatest Christmas gift I've ever given anybody. I just cartwheeled down the corridor. I told my wife how pleased I was as I'm called last night. and you know, this is the real and palpable outcome of years and years of just starting out with young researchers, driving them and supporting to the system, and then we get these outcomes. And you know, stroke has just been magical, and I'm just looking forward to the next areas of Parkinson's and Alzheimer's and things like that. And so I just want to finish by thanking our speakers, but thanking everybody here who's contributed to tonight, the staff, and also everybody who's coming along to join in and as supporters of the Foundation. So thank Richard and Alan, and thank yourselves. Yeah. I repeat my reminder that only paid up members can vote. And I'll start with noting a couple of apologies. Aidan Hewitt, who's our Deputy Chair, and John Burton, who sent apologies, previous Chair of the Foundation. We try not to print too much paper. The minutes of the past meeting are available on our website, and I want to start out by confirming those minutes for those who have a look at those and remember and would like to move that we accept those minutes. Second. Deb, all those in favour? Thank you. Now, any matters arising from the minutes that anybody wants to bring up? We'll just come back to the SGM in a moment. So, briefly, I want to now do my report. This has been another really good year for the Foundation. Rich will go over the amounts of money and things like that, but it's really been a transformation. And I alluded to this briefly before. Just in the last few years, it's really come to our attention and pretty much forced on us that the Foundation had to change. We had a rather old-fashioned logo. We had an old-fashioned way of doing things. We had an old-fashioned governance structure, which for many years was fit for purpose. And we can look back with great pride for all the things we've achieved. But gee, the world is changing really, really fast. A large proportion of the donations we receive are still in cheques. I don't own a chequebook. And Kiwibank is about to pull cheques. So there's a lot of things that we have to change in this modern world. The donors have been terrific supporters of the Foundation, but we felt that we're probably losing touch with the next generation of people interested in neurological disease and becoming potential donors. The world of governance has also changed as well. The Charities Commission is taking a good, hard look at organisations just like us, and we know they have looked at us, and they look at all charities now. And we have to have proper governance processes in place, and we have to have proper systems, and the Charities Act is changing as we watch. And so we had a lot to do in the last 2 or 3 years, and I'm pleased to sit here and say that we've done a lot of that, and we're moving in the right direction. We've changed the internal committee process in the Foundation. We've improved our audit and compliance, and we get good ticks from that from our auditor. We have a much more transparent system. We've developed a values-based system that we're following better, and we have getting professional help to do things such as our HR. So the Foundation itself running on the systems and science working so much better. We've also worked very hard as a team, but also with specific specialists to improve our our outward facing, the way we communicate with the community. Just launched our new website, and I urge you to have a look at that. We've changed our logo, we've changed our colour, which some of the oldies didn't like that much, so but there you go. And we've done this with communication with the community, and it's been really, really interesting. So those of you who remember the old logo of the foundation had a profile head with a smaller head underneath, which kind of looked to us like somebody growing brains. But interesting, quite a few people in our focus groups looked a little bit paternalistic and a little bit dominating and a bit threatening. Go figure. And so it taught us once again that you just got to go out and communicate and talk with the community. We've had a really busy year of community talks and awareness raising. And I just want to remind everybody here that it's actually part of our deed that we do awareness as well as research. And we recognise that we're probably not presenting our finances well enough to indicate that, and because we're trying to put quite a lot of our resource in a balanced way into making sure that we get out to communities and improve awareness of brain. And that's got really important health implications at a couple of levels. Personally, at a personal level, Alan pointed out before that a third of the people with these strokes that can be substantially reduced if you take special sorts of anticoagulants, but we're not getting that message into the community properly, and we're getting support to do that better. So at a personal health level, awareness makes a big difference. At another level, it's also important because we need awareness of neurological disease to come up in parallel things like cancer, child health, cardiac disease, which are all important, but neurological disease has often been the poor cousin in the relationship, and the awareness raising that the foundation does helps pull it up into a higher scale and get the ear of the politicians and the funders and the hospital boards and things like that. Rich, I think, is going to talk a little bit about some of the people changes in the foundation. I'll mention that. And we really have had a whole change, and it's really thrilling to go to the foundation now to see a really young group of people. It makes me feel old, but attacking the issues and the problems with great enthusiasm. So sort of from top to bottom, inside out, I can say that we've had a really good year, and I'm really proud to be your chairman, and eternally grateful the way the foundation's changed my life in all sorts of ways, except probably Christmas Eve. But thank you all. So there's the chairman's report. Rich.
Thank you, Barry. So for those of you that haven't met me, Richard Easton, the CEO of the Neurological Foundation. Been here just over 2 years now, and as Barry's talked about, seen a world of change. So my role tonight is to present you a brief summary of our financial report, and this is built on the back of the formal audit that we went through at the end of our financial year with our auditors, Deloitte. So our financial year is 1st of April through to 31st of March, and the report is for the 31st of March ending in this year. A really strong year for us, and in particular a huge thank you to all of our supporters who have enabled us to be able to fund so much research during the year. We funded during the year 45 new pieces of research, but just to give you some example, there were over 100 applications during that same time, and many of those were good quality pieces of research that we just weren't able to fund, simply because there's only so much that we're able to do. And that's enabled something more than 19 new researchers directly or indirectly working across teams in the laboratories across the whole country being able to work on the research. That's a really brief summary for during the year, for that financial year, donations from requests and from donations from all of you, totaling just over $7 million, of which nearly $4 million was through donations and just over $3 million were through requests that we received through a variety of really wonderful people that had taken a long-term view of being able to support us even after they passed away. And that's a huge thank you from us. It's really important. We can't do the work that we do. We don't get any funding from the government. Government funds other areas through such things as Richard was talking about, centres of research excellence like Brain Research New Zealand and the Health Research Council. we're the only organisation in New Zealand that actually funds research, specifically with a neurological perspective on it. We also have an investment portfolio, so gradually over the years where people have been generous, we're making sure that we've invested wisely, because as you've heard tonight from both Sir Richard and Alan, the research isn't something that happens overnight in 1 week. Actually, it takes 20, 30, 40 years. And we know that some years the income we get is wonderful, and some years it's just a hard year. There are challenges going on in the economic sector, and so making sure that we're actually able to sustain and guarantee research, even if it's been a hard year. And last year we had about $3 million of income through our investment portfolio, which meant that we could guarantee that the researchers were still able to continue the work that they do. And our costs have changed slightly. The council at the beginning of last year gave us some commitment as an organisation to actually change the things that we're doing. So as an organisation, we're trying to make sure that we sustain and continue to do all of the wonderful things that we have always done for the last 40 plus years, but at the same time make sure that we reinvent ourselves a little bit, because one of our challenges, we have a really wonderful supporter base, but many of them are getting older and sadly, as we've seen through the requests that we've been receiving, passing away, and if we don't actually reinvent ourselves, the ability for us to continue to fund research in the future will disappear. So looking at, through the work that Barry mentioned earlier, at our website, through looking at new areas that Kerry and her team have been driving around community and peer-to-peer fundraising, getting people more involved in, not always regular giving, but looking at new ways of giving. And then looking at how do we significantly ramp up the education awareness and the communication through headlines, through events that we've been doing around the country. In March this year, during our annual appeal week, we had over 20 events across the country. We introduced a Women in Neuroscience series of events back in February this year. So really trying to actually take research and researchers like Alan and Richard out into the community, because we know and we've seen how valuable people being able to talk and share the passion that they have for research is in helping people to understand money that they've donated to us is actually going to a really good cause, and that there is a pathway to hope for some of our work. So a real brief summary, the appropriation to research included in our audited report, the renewal of our relationship with the Chair of Clinical Neurology, which is for 5 years, and the renewal of the Brain Bank for 5 years as well. So overall our appropriation to research is $6 million. And then underneath that our costs, so that education and awareness program was almost $500,000 that we put in to making sure that we took researchers that are in the country. Our marketing, which includes our website, includes headlines, includes all of the other communication we do, was about $307,000. The fundraising, so Kerry and her team are actually actively spending time out in the community, talking to our supporters, helping to make sure we understand their needs. We have two of them here tonight, Kelly and Margie, really making sure that we understand what people are looking for when they donate money to us, and the cost of that business, including our database and the database tool and the team that are needed to support that. It's about $800,000. And then our operating costs are running the team of people, running the buildings that we have and the vehicles, and their ability to actually post out receipts to people, etc. It's just over $1 million, so that's a brief summary. And look, we're really conscious every dollar that people give us is really important, that we find ways to be able to connect that into research. So we're spending some time at the Audit and Risk Committee just recently starting to think about how do we make sure that people really can connect the money that they've given us to the outcomes, and over the next couple of years as a charity, we're a tier 2 charity, and so not this financial year or next but the year after, we're actually required to be audited not just on our financial outcomes but also on our service outcomes, and so we're just starting some work right now to make sure that we can actually tell you a bit of a story about that. So hopefully most of you will have seen the wonderful work that Danielle and her team have done around the report to members. Over the next couple of years, you'll see that evolve a little bit more about being able to tell a story of not only what income did we receive, but where did it go to and how did we use your money as well. So that's a brief summary of our financials for you.
So any questions from the chair or the CEO report? Okay, so I'd like to move that both reports are accepted. Do I have a seconder? Kathy? Yeah. All those in favour? Against? Now next we move on to election of council members. I'm going to I'll normally pass over to Barber and the deputy to do this because I'm one of the re-elections, but Tim Anderson, Professor Anderson is one of the councillors has agreed to lead this for me. But before we do, I just want to acknowledge Tony Offen, who is Tony, who's stepping down this cycle. Tony from Dunedin, and has really been just a major force in the council over 16 years, Tony, yeah. And so really, Tony has seen the organisation evolve from, I think, a 10 million organisation to a 70 million organisation. He's been a major force in leading us with our governance, leading us with our financial understanding, and just a really wise head. Has been an important supporter in Dunedin and helped drive through the Professor of Neurosurgical Chair, and also just as personally as the chair, I found a great comfort in council as his time as deputy chair before he handed over to Barber, as just a really sensible person. So on a personal level and on behalf of council tonight, I'd like to thank you very much for your time with us tonight. Just for due process, he's going to resign right now, then we're going to co-opt him back on council tomorrow to do his report. It will follow due process. Yeah.
Okay, thanks, Barry. So I'd like to propose 3 existing members to be renewed onto the board. Those are Deb Peters, Barry Snow and Wayne Boyd. As Barry's mentioned, I'd like to formally sort of announce the retirement from the council of Tony Hoffman. And very pleased to propose 2 new members, Sonia Clapham and John Kelly, for election onto the council. Further, I'd like to, if I can carry on, continue the nominations. I propose re-election for council chair of Barry Snow, and also the re-election of the council deputy chair, Bailey Muir, who's not here. So those are the nominations for the council.
So you got a second? You made the nomination? Yep. So who's second?
I made the proposal.
Yep. And you want to call for a vote?
Yeah. All right. Okay. Yeah. So all those who are in favour of those members and new members, say aye.
Aye.
Any against? Seems to be carried unanimously.
Thank you, Tim. It's a relief. Got a bit of skin in the game. Okay. Now, next part of the standard business is to reappointment of the auditor, and we'd like to propose that we reappoint Deloitte as the auditor for 2019-20. That's the proposal. Do I have a second of this? James? All those in favour?
Aye.
Against? Passed. Next is the report from the SAC and the PAC. Kathy, you're going to lead us out?
Yes.
Yep.
Do this up here.
And Bronwyn here.
Thanks, Barry. Hello, everyone. My name's Kathy Smear. I'm the departing chair of the Scientific Advisory Committee. It's my pleasure to just give you a brief update on some of the activities in the Scientific Advisory Committee over the last 4 years that I've been in the chair. And then I'm going to hand over to Dr. Bronwyn Kibble from Victoria University Wellington, who is one of the new chairs who will be taking them into the future. So I'm very pleased to report that the research funding application processes used by the Foundation have been substantially improved over the last little while in several important ways. For example, we have improved our management of conflicts of interest, where members of the Scientific Advisory Committee who are named applicants on an application are stood down for that funding round and play no part whatsoever. The New Zealand neurological research community is very small, and so it can be tricky to find enough people with enough expertise to review applications in depth. But we're fortunate, and I think it's in no small part due to the Foundation's work, that the workforce has actually grown quite a lot over the last 10 to 20 years. And so we're lucky that we now have a pool of expert committee members. So instead of having the same sort of standing group of about a dozen, we now have just over 30 expert committee members. And these people can be drawn on for each committee round, depending on their expertise and how that lines up with the types of applications received. And we can comprise that committee each time, ensuring some diversity as well, in terms of gender, in terms of career stage, institution, so we get a really good mix of perspectives around the table. It's really healthy. So our hope is that instead of having sort of a dozen standing committee members who stay on and stay on and stay on, and we sort of wear them out completely and then they leave, instead of that sort of conveyor belt, we're hoping for a revolving door where we have this pool of experts who can come into the committee as they're needed and then move out if they apply, because then they can't be part of the process, or if perhaps their expertise doesn't line up that well with the applications in that round. So we're really excited about this new approach. The Scientific Advisory Committee primarily looks at applications for funding for projects, as you've heard earlier this evening. We also have a new committee called the Personal Awards Committee, or the PAC, and they look after applications for scholarship and fellowship funding. So this is for the researcher as opposed to a project being done by several researchers usually together. So this new committee actually has quite a large undertaking because we've developed several new fellowships to better meet the needs of the research community. We spent a lot of time last year in particular going out to the research community and really finding out from them what are your needs in terms of research funding. And we found out that we were doing a good job, but we could do a better job. And so we now have some new fellowships available. And so the role of the PAC has grown considerably because there are more funding streams for people to apply into. We've also had a switch, which we think is environmentally sound, where before all of our applicants for scholarships and fellowships would actually come to Auckland if they were in the country so that we could interview them in person. We're now doing all interviews by video conference to minimise our carbon footprint, and that includes people who are just sort of 300 metres away, just in the next building, still do it by video conference so that it's fair for all applicants. Both the Scientific Advisory Committee and the new Personal Awards Committee, each of those committees actually has 2 co-chairs to spread the workload because they are all volunteers, they're all very busy people, and also create some redundancy so that if one of those chairs has an application in the round, the other person can step in and manage that round. There have been several other improvements, which I won't bore you with the details, into our processes, primarily aimed at improving confidentiality as well as a new scoring system that the PAC will use to improve fairness and also transparency of its decision making. It's been a real pleasure for me to be part of the Scientific Advisory Committee for the last decade and to chair this committee for the last 4 years. And I thank the Foundation for entrusting me with the responsibility of reviewing our research and investment strategy and leading the improvements that I've outlined here. It's been a huge privilege for me to do this work. I've now handed over to 4 new co-chairs. It's not lost on me, I'm being replaced by 4 people. The job has grown enormously in my time. And I've got every confidence in them and our new research manager, Dr. Sarah Schomburger, that the Foundation will continue to identify and fund the best quality research in New Zealand to fulfil its mission and its promise to our supporters. It's been a great privilege. Thank you very much.
Before we hear from Barber, I just want to thank you on behalf of the Foundation, Kathy, for all the work you've done. You've completely transformed the way this does. You've put a lot of kind of thought to that, and it is definitely a much better process from when I was chair. And I just want to thank you for the amount of work. If Kathy was on the committee for about 10 years, she probably read about this much of grant applications if you pile them up in paper. So that's about 40 warm pieces. Yeah, so thank you. We really appreciate it.
And this is Bronwyn, sorry. Hi, I'm Bronwyn, and I'm from Victoria University here in Wellington, and I've got to say thank you, Kathy, for all of your work, and I can't believe that you did it all by yourself, and I'm really, really glad there's now 4 of us to share the workload, because I feel confident with the workload shared that we have got the time to put in to really rigorously assess all of the science that comes in to make sure we do fund the highest quality work and research. And all the people on the panel, they do it voluntarily, and they do have big piles of paperwork to read, and they really, really do put in a lot of work at these meetings to really make sure it's a really rigorous and fair process. And I think of all the committees I've been on for grant funding, I think this is one of the best, and it's because of all the hard work and all of the changes that have happened fairly recently, by it sounds, but I'm really happy with the place that's been handed over to us. So I'm really happy that, I'm hoping that we don't break it. And I think that was advice that you gave us, Kathy. Just don't break it. No, I'm sure we won't. And I think it's a trend that more and more applications are being received. The neuroscience community in New Zealand is growing, research is growing. We received 43 applications in the last round, and the cost of the applications were totaling just over $4.9 million. And you cannot fund all of this research, even though most of it is very high quality. So the SAC committee rigorously evaluated these with external reviewers, and there were 24 large projects that were submitted, and we're recommending that Council will approve 5 of them, and that's over $1 million of funding of those projects. There was also 13 small projects, and we would like to recommend that 4 of those will be funded as well. And that basically takes it to all of the funding line of just under 1.2 million for this round. So that's a lot of money going into research. It's a lot of high quality work, and it's really exciting to be part of it. And it's not like we don't get a lot out of this as well. We read these applications, and we're really, really learn from reading these applications and the quality of the science, and just nurturing our ability to write grants in future. You learn, oh, that's a really nice way to say that. So it's building the people on the team, the SAC. There's a lot of young people on the team as well, and I think it really does nurture the people coming through. By being on the committees, you see some of these grants and go, wow, that's, and you learn from it. So it's a big learning process, and it's really, really exciting to be part of that. And I'm really glad that there's 4 of us doing your job, Kathy, because I think you can manage the workload.
Thank you. Yeah, thank you. Okay, so we're almost at the end of our meeting. Just general business. There's one general business item that I want to raise, was to remind you that we had a special general meeting to a couple of months ago now, where we revised the rules of the foundation. I assure you that we didn't change any of the core founding principles of the foundation. The majority of the changes were really to bring it up to date. The document that we were dealing with was typewritten and didn't have anything about email or anything like that in it, so we had to make those sorts of changes. We also had to make changes to bring it up to modern rules about running a charitable organisation. But probably the most important thing that we changed is that we've got rid of what was very long tenures, which is no longer acceptable good governance. So we have to down to part 2 of 4-year tenures, and then you have to rotate off the council. We thought about it very deeply, what we do with all the long-tooth people who've been here on the council for quite some time, maybe. And we didn't want to do a clean sweep by changing the rules, because it would have meant the chunk came out. So we've taken the policy of replacing, or bringing on 2 new council members per year. And Sonia and John, we're really pleased to have you on the council. Kind of new figures, new faces, new ways of thinking about things. And we're going to keep this going for some time to keep it all refreshed and connected to the community. And so that's all. So is there any more other items that anybody, any other member of the
Councillor's satisfaction, and I'll therefore call this meeting to a close.